My name is Farr Curlin, and I am the faculty director for the Arate Initiative, an initiative in the Keenan Institute for Ethics that seeks to promote research and educational opportunities that focus on the virtues and the shape of a worthy life, primarily in the workplace, in the public square, and in the university. And tonight we're delighted to welcome you to this lecture by William Hurlbut. I want to say, before we begin, thank you to our co-sponsors, the Trent Center for Bioethics, Humanities, and the History of Medicine, and the University Program in Genetics and Genomics. This is an important topic. The Mueller report has somewhat displaced it from the headlines, but very recently, as many of you know, the news cycle was dominated by reports of new ventures with CRISPR-Cas9 technology, which I know Dr. Hurlbut's going to speak about. Tonight, Dr. Hurlbut will lecture, and then we're going to have some time for Q&A. And to introduce our speaker and to moderate that Q&A, I'm glad to welcome to the podium Charlie Gersbach, the Rooney Family Associate Professor of Biomedical Engineering.
Thank you very much. It's with great pleasure I get to introduce Dr. Hurlbut for his lecture. As someone who's involved on the scientific side of genome editing, we've obviously been paying attention with great interest to everything going on in the news and in this general area, and reading everything that Dr. Hurlbut and his colleagues have to say about the ethical side of these issues. And so I'm happy that we'll be able to hear more about that in person today. William Hurlbut is a physician and adjunct professor in the Department of Neurobiology at Stanford University Medical Center. His primary areas of interest involve the ethical issues associated with advancing biomedical technology, the biological basis of moral awareness, and studies in the integration of theology and the philosophy of biology. In addition to teaching at Stanford, he's worked with NASA on projects in astrobiology and as a member of the Chemical and Biological Warfare Working Group at Stanford's Center for International Security and Cooperation. He has organized and co-chaired 3 multi-year interdisciplinary faculty projects at Stanford, including Becoming Human, the Evolutionary Origins of Spiritual, Religious, and Moral Awareness, Brain, Mind, and Emergence, and the ongoing The Boundaries of Humanity, Humans, Animals, and Machines in the Age of Biotechnology. In addition, he is co-leader, together with UC Berkeley professor Jennifer Doudna, of a new initiative entitled The Challenge and Opportunity of Gene Editing, a project for reflection, deliberation, and education. From 2002 to 2009, Dr. Hurlbut served on the President's Council on Bioethics. Please join me in welcoming Professor Hurlbut.
Thank you for that nice introduction. I don't know about the politics. I was thinking you guys might be preoccupied with basketball. So we have amazing technology to talk about today, extremely important and worthy of some time. A couple of years ago, I was presenter at a conference at Harvard entitled Editorial Aspirations, and I thought it would be interesting to start with a picture of their flyer, because this was human hands, what Aristotle called the tool of tools, the symbol of our distinctive body form and unique capacities of mind, our comprehension, creativity, and control over the world in which we dwell. Now it's turning more to capacity to control our actual bodily selves. Nowhere is the power of our biotechnology more dramatic than in, currently anyway, than in genetic engineering. So what I want to do actually is go a little too fast through what I've presented, because I think it'd be more fun for all of us if Charlie and I get up here and answer some questions and have a dialogue. So excuse me if I go a little fast, but I'll try to go deep as well. So 70 years ago, do I need to speak louder? No. 70 years ago, Aldous Huxley, anticipating the transformation of human life through advances in biology as the final and most searching revolution, asserted this really revolutionary revolution is to be achieved not in the external world, but in the souls and flesh of human beings. Now, with CRISPR-Cas9 and related technologies, and there are quite a few of them too, we're at a hinge point of this prophetic vision, poised for a significant acceleration in the momentum of advance in biotechnology. This holds great promise for urgent and uncontroversial progress in biomedicine, agriculture, and environmental ecology, but it opens fundamental questions about our place within the natural world and the uses of these technologies in shaping the human future. So MIT Tech Review has called CRISPR the biggest biotech discovery of the century. CRISPR is an acronym. I'll let you read it. So it's adapted from an immune system deployed by bacteria to protect themselves against viruses. And it's been described as a genetic scalpel or molecular scissors, but it's more rightly compared to a Swiss Army knife. It's composed of two basic, it's multi-dimensional, it could do many things. So there's the Swiss Army knife. It's, just to give you a little basic science before we start here on the ethics, it has two basic components. A guide RNA sequence that identifies and attaches to a specific stretch of DNA, and an endonuclease, an enzyme which, in one form anyway, cuts the DNA, allowing a range of operations on the gene segment, but it can actually attach other functional dimensions. So scientists are exploring CRISPR and the many natural variations and selectively engineering clever new modifications in its targeting and operation. And they are developing a whole new genetic toolkit for operations at every level of genomic process. The breadth, flexibility, and precision of these new tools is opening a vast increase in the range and complexity of experimental possibilities, theoretical insights, and practical applications. My co-leader in the project we're doing through Berkeley has made some very strong statements about this. I'll let you read those. But what can this be used for? Well, just think of anything that has DNA in it. You pretty much got it. More nutritious fruits, new forms of photosynthesis, programming operations on different levels of genomic expression, like RNA as well. There are just so many things. It's really astonishing. The individual genes can be strategically disrupted or selectively altered to study development of disease. New genes can be inserted. Whole patterns of gene action can be simultaneously studied, and this will allow us to create new drugs, new farm animals, all sorts of things. It will also allow us to study human development, and a range of very important insights can come from these technologies. Together with deepening knowledge of genomics and our exponential increase in gene sequencing capacities, these new tools for understanding and control the most basic elements herald revolutionary advances in biotechnology. I was at Stanford as a medical student when Paul Berg more or less brought the idea of gene editing, of recombinant DNA on the map. And it now, in the last 5 years since CRISPR was discovered, it feels like finally, after almost 50 years, that the technology is coming online in a really effective way, opening up vast new possibilities. In the few years since its introduction, CRISPR-Cas9 and related technologies has been employed in labs worldwide. Billions of dollars has been invested. The scope and versatility of this technology promises transformational impact as great as the discovery of electricity, synthetic chemistry, and nuclear physics. I think it's just the next step in the great story of the human technology. While much of this advance is welcome and constructive disruption, as we say in Silicon Valley, providing urgent and uncontroversial progress, there's also many troubling and potentially worrisome dimensions of this technology? Well, it can be used to study human development. It can be used to modify animals. This is a long time ago, actually, but gives you an idea. I think it's possible that our grandchildren, or maybe our children, are going to go to the zoo and see genetically modified animals. Is that a little too far out, Charlie? Or it's just amazing what you could do, and especially when the animals don't have to survive in a natural environment, you could create variations that nature couldn't select for. You can create human-animal chimeras. Don't worry, this is just a drawing. But it does raise interesting questions and ethical questions about combining human and animal genes. So, as my colleague at Stanford, Stanley Key, says, we should think carefully about how we're going to use this. There are serious ideas suggested already for things that are intrinsically edgy in the sense that we want to be careful with them. Major levels of ecological engineering. As one Harvard postdoc said, we need to correct the fact that nature is fundamentally amoral. Well, I don't know about that. Gene drives that open the possibility of changing whole species genetics very quickly. These are mosquitoes. We could potentially eliminate the mosquitoes that carry malaria from certain areas or diminish their numbers, but could we destroy or unbalance whole ecological systems? Moreover, there are suggestions by people like George Church at Harvard for the de-extinction of our ancestral species, Neanderthals, Denisovans, as well as calls for genetically engineering potential astronauts to go off into space. Well, these are all far out ideas. Here's a really good one, downsizing. Maybe we could create human beings that are a lot smaller, we wouldn't use so much resources. That's, of course, a joke. But nonetheless, there are very, very serious people talking about our post-human future, people calling for interventions to extend human life great lengths. Most specifically, the transhumanists, who they're an interesting group. I don't know if you have them here at Duke, but at Stanford, the transhumanists is a growing sort of cult club at Stanford, and they look back at evolutionary process and they see it's cruel and contingent, and basically they say we can do better, and so they suggest using our genetics and our AI technologies to create human-machine cyborg post-humans. I think maybe they'll end up with the Darwin Awards eventually, but anyway, so I want to lay out just a few prospects in this to set the stage for a discussion. How human genome editing might be used, what biomedical research uses there might be, and also our visions of the future. Because overarching all of this is a sense of what is our role? What is our legitimate role in the natural world? And what is our wisdom that will guide it? So this is a picture of Jennifer Doudna. She and a French scientist named Emmanuel Charpentier co-discovered CRISPR-Cas9, which set the opening for the whole CRISPR advances. And she is very excited about it, but she's also quite worried about where it might go. And that's obviously very uncontroversial things. For example, for the most part, if you're going to intervene in an existing patient, it's a matter of safety and medical concerns that are typical of many other advancing technologies. In this case, sickle cell anemia, if you could take the way they would do this is they'd remove some of the blood, alter the blood-forming elements of the bone marrow, that the hematopoietic stem cells, and put them back in and have them continuously generate repaired, properly functioning red blood cells. Without that, they turn into little sickles and they form clumps in the arteries. And when I was a medical student, I took care of a little boy who was 13 years old, and he'd been hospitalized 200 times. So this would be an extraordinarily wonderful advance and probably pretty uncontroversial. There are ways to target this, not just working on cells in the lab, but target sending CRISPR-Cas9 into the body. It's targeted to specific cells, perhaps even brain cells. And so that too would be potentially very, very beneficial therapy. And then, of course, the question comes, well, if we could treat diseases this way, what about preventing their development in the first place? And that is a really profound question because it opens the issue of germline genetic engineering, going to the bottom of the beginning of a person, influencing not just their existing body cells, but the very germline cells that go on to produce their own offspring. There are many, many genetic diseases. There are at least 6,500 single gene genetic diseases. There are probably tens of thousands of them that we haven't yet discovered. And 95% of the known genetic diseases, single genes, have no treatments, no cure. And some of them are really, truly horrible. This is a picture of a little boy's hand. He has Lesch-Nyhan syndrome, and these little children will actually chew their fingers, chew their lips off, and it's just terrible. But that, of course, raises the strange question of what risks would you take? And there are many questions. We can talk about those later. But the strangest of all is what if people want to do more than just treat the obvious severe diseases? So is albinism a disease? I mean, a person with albinism lives perfectly decent life, a little socially awkward maybe at times. Who knows? I mean, people feel socially awkward for all sorts of things. And yet, they have trouble in the sunlight, but they can live normal life. But other people say, well, why not? Why shouldn't parents be able to make the kind of children they want? It's always blue eyes, so I put some brown eyes too. But what about if we figure out some of the basic neurobiology of things like dyslexia, and there's genetic components to dyslexia for sure. And is that a disorder or it's a disease? Maybe it makes it hard in school, but they're also great strengths. You may not know this. Charles Schwab has dyslexia, so does the guy Bronson who started Virgin Atlantic, and a lot of famous people. And beyond that, George Church at Harvard has identified what he rare protective variants. He's got a list of these these are disorders that are, or circumstances. They're not disorders at all. They're just alleles. They're forms of a gene that is not a disease-causing gene itself, but is associated with a better outcome or less vulnerability to certain disorders. And he says that if these were not rare, if they were more common in the human population, then we could have significantly better general health. And among these, a whole variety of things: less sensitivity to pain, leaner muscles, stronger bones. One of them down here somewhere says low odor production. I'm not sure what that's about. Anyway, so what about that? Are these emerging positives in the human gene pool, or are they balances against something good and bad? I was very good friends with Baruch Blumberg, who got the Nobel Prize for discovering hepatitis B. And he used to say to me, there are no, apart from really severe diseases, he meant, there are no good genes, bad genes. There's a balance of what the body needs to contend with. So this raises some very interesting questions about what we would do if we could intervene. And developmental biologist Stuart Newman warns us that opening the door to attempts to pick and choose characteristics, well, who's going to say what counts? It also opens the possibility that for misuse in a negative way, by judging people, defining diseases is often very socially constructed. And for example, this disease, anybody know what that disease is? That's called drapetomania. It was a disease outlined in the medical textbooks of the antebellum South, and the name is formed from the two Greek words meaning a tendency to run away. So this poor fellow was whipped. That was his cure. And of course, we all know the horrible story behind this, and the way it ended up was incredibly tragic. This is genetics misunderstood, but misapplied as well as misunderstood. In fact, genetics is not nearly as determinative as most people think it is. Studies of twins do show some concordance, but it's what's remarkable is how different twins actually are. Nonetheless, I think we have to take seriously the fact that we're at a new era, that there's a new challenge and new possibilities. Not only might we use these to intervene in serious diseases for both somatic level and germline level, but there will be calls to use it for improvement, not just at the germline level for making better humans, but at the level of up and running people by altering regulatory changes, affecting regulatory changes, and that too poses many ethical questions. So we need to pause and ask ourselves, well, what exactly is it that we do as human beings? What are we in charge of? What's reasonable? And particularly as physicians, what are our medical standards? And by what principles will we operate in this realm? It's clear that driven by appetite and ambition, humans are a pretty open-ended species. They want what they want. I mean, that's probably pretty common understanding in the humanities anyway. The danger is that now we have power to get what we want more than we ever did before. And increasingly medicine is being defined by a new paradigm rather than what Galen said, the healing. It's being defined by the paradigm of liberation, and liberation from all that's unattractive, imperfect, or just inconvenient. And look at the way medicine is already drifting along. And I'm not criticizing these things. It's just warnings to us about where we could go and where we want to go. Rogaine curing, when I was trained in medical school at Stanford, male pattern baldness was not considered a disease. By the time I got on the President's Council on Bioethics, it was a $2 billion industry treating male pattern baldness. At the same time, I did a little research at that point and figured out that even though we were spending $2 billion treating baldness, the whole world collective effort and research to address the problem of malaria was a mere $100 million, 1/20th. So it's a little warning to us. Nonetheless, there are things that we might intervene in. Short stature is socially difficult, so we got growth hormone, and then of course we've got pills for everything now. Actually, we don't yet. I mean, we have our pharmaceuticals target maybe 200 protein sites. That's what I've been told anyway. And there are probably thousands of protein sites that could be targeted in many other ways to intervene too. So we're looking at a multi-generational advance in interventions against very serious diseases, but also with the knowledge that could be applied to advancing purposes for which medicine has not traditionally been considered applied. And then, of course, you wonder how far it might go. I think it won't go as far as people think it will. Better brains, genetic alterations, the Immortality Institute. But nonetheless, William Haseltine said the real goal is to keep people alive forever. You know, one thing I should mention as I go here is that genetics is much more complicated than most people and most lay people realize. Genes, there are very few genes that have a very direct effect on a trait, a single gene and a single trait. Red hair is one that's really, is that right, Charlie? Red hair is, I think red hair, you can get red hair with an alteration of one gene. But most traits are the result of what they call polygenic inheritance, with many traits bringing about the gene, and most genes affect many traits. So it's a little bit like, genes are a little bit like colors on an artist's palette. It puts down the primary colors, but before they make it to the canvas, they're mixed. So anything on the canvas is a mix of many genes, and many colors, all the colors are used for many different things in the canvas, if you get the analogy. But nonetheless, I think we need to take seriously the moment we are in. It's no longer science fiction. The powers are coming for alterations of human life that we've never really thoughtfully, we've never encountered and thought through carefully. It's kind of a high-tech world. And recognizing this coming problem, especially to germline genetic engineering, Jennifer Doudna and several Nobel laureates, Paul Berg and David Baltimore and so forth, called for a gene editing summit in December 2018. They met in Washington and issued a statement strongly cautioning, not calling for a moratorium, but a worldwide consensus, basically, that we should not proceed into germline genetic engineering. And that there has been studies done on basic embryonic studies of putting CRISPR into human embryos. That one of those happened just before the gene editing summit. But for the most part, there's been very cautious, stepwise investigation of these gene editing tools because there are certain downsides associated with them, including off-target effects, producing potentially dangerous editings. And they were getting closer and closer. But in November of last year, the second gene editing summit was held in Hong Kong, and both Charlie and I were there. And just on the eve of this summit, as most of you probably know, it was announced that this young Chinese scientist, Dr. He Xiongqi, known to his people who know him, call him JK for short, so I'll refer to him as JK. JK announced, well, it was actually leaked. He didn't want to announce it, but it was leaked that he had performed germline genetic engineering and 2 little twins had been born. And in this case, he had altered a gene called CCR5, which it has been seen primarily in European populations to confer degree of protection against HIV. This is a picture of HIV attacking a cell. The yellow is the virus. And the virus enters through a little portal, and if you can disrupt that little entrance gate, you can prevent it from getting into the cells. And that was what he did, and he produced these 2 twins, so a pair of twins. Well, that raises a whole new prospect and was, as you know, widely condemned. And I want to talk to you about that more because I know JK quite well personally, and it's very interesting and complicated issues. But let's do that in the discussion section. I want to go on and lay out some more of the issues associated with this. So in the meantime, before this had happened, there was quite a lot of call for research on human embryos because, as Paul Berg and David Baltimore said, we can learn a great deal by investigating the most basic elements of living systems. And that was the embryo study that had already been done. George Daly, Dean of Harvard Medical School, said gene editing could be used to engineer specific disease-related mutations in embryos, which should produce models and be very useful in terms of scientific knowledge. So this brings us back to the issue that those of you who are at least at a certain age will remember was a very deeply contentious issue in the United States. And this was the years I served on the President's Council on Bioethics. And I had a front row seat on this, both because I was serving on the council and because I made a suggestion during those years to try to bypass the conflict to meet the good goals of both sides. Because really both sides were protecting something important. On the one hand was advances in science. On the other hand was concerns for human dignity, lest we use developing human life as a raw material in a project of scientific advance. So both sides were making a good argument. I thought there would be ways to bypass it, and so I made a suggestion known as altered nuclear transfer, but within a few years, and Rudolf Jenisch proved it could be done in mice, and we were ramping up to do it. We did it with primate cells. We were ramping up to do it with humans, but in the meantime, the Japanese scientist Shinya Yamanaka figured out a way to reprogram cells, and that's what we know as induced pluripotent stem cells today. And just a little side note on this, I worked very hard to get our country to seek an alternative, and it was very, very challenging to get the government leaders to discuss the possibility of putting a little bit of money into figuring out an answer to this. I spent many days and a lot of my own money communicating with people and traveling to Washington to talk. I met with quite a few senators and congressmen. Finally, we got a bill to the floor that would just put $25 million into this, which is, you know, when they say in Washington, it's a rounding error. But it passed 100-0 in the Senate, and then it didn't pass in the House. The contention was so great. And then a year later, President Bush signed an executive order to fund it. But in the meantime, for a mere $100,000 of private money, Shinya Yamanaka solved this problem. And to me, I just want to mention that because I think there's solutions to a lot of the problems that we face as a society and will be with the biomedical technology if we just are creative. But if we're going to just fight about basic things, we will put off advances in science. And nonetheless, I think we have to take very seriously the core principles that are being fought about. And that is going to become more and more essential because even as human embryos were a point of major contention in 2001 before 9/11, it was the subject of the first address by President Bush to the nation. That's how serious it was. And those of you who were there remember that it was very deep. It was as though suffering on the one hand and human dignity on the other were being played against each other. My own medical school had this cover on my alumni magazine. I don't think it was very fair. It posed religion against science. Was not the truth. There were sensible core principles of concern on both sides of that equation. The concern for human embryos was expressed in 1995 in the Dickey-Wicker Amendment, and that's what the contention was about, because it's illegal to fund embryo research, it's not absolutely illegal, it's anything that endangers, destroys, or creates for instrumental use human embryos. And that's not encoded in law, but it's renewed every year as the Dickey-Wicker Amendment. And that's what was being discussed. And during those years, it was possible to do research on human embryos with private funds or state funds, which is why California passed a $3 billion stem cell initiative to fund that. But this was a very difficult debate. And I want to talk about germline genetic engineering in the discussion, but I want to raise an issue that's not being raised much yet, because it really is, to my mind, a much more potentially contentious issue in our society. I don't actually think CRISPR and the advances in gene editing and the controversy over germline editing are going to be the deepest and most difficult fight of this issue. And even if they do happen, I think it'll be fairly marginal, although I want to talk to you about the potential dangers of commercial application of these technologies. But what is undeniable, I think, is that the uses of germline genetic engineering for the study of developmental biology is very real. Now, these are embryos that may never end up implanted in a woman's womb, but they nonetheless will develop in the dish. And what you could learn, knocking out genes and seeing what the genes did, or putting alternate forms of genes into embryos and so forth, is very real. And I think that's what is not yet hit the radar in the general public discussion, but I think it's going to get there. I can hear it already in people I'm talking to in Washington and so forth. So I want to take you on a little bit of a trip down that journey and then wrap up and talk together. So this was the scene at the time. We fight because lives can't wait. And of course, both sides were fighting for life. That was the strange thing of the. And this is a political ad. Now, before I show you this, I want to make the point that what was going on in the embryonic stem cell debates was on the one hand was human disease and the suffering, very real suffering, and the other hand was developing human life. And so you can imagine that if that was the argument was going on over embryonic stem cells, all the more will go on with CRISPR, because the modification of embryos to study the formation of disease, natural health, and a whole range of other things I will lay out in a minute, has a promise, the scientific promise, of far more than it encompasses and extends the whole argument that was made for overriding our nation's principle of the Dickey-Wicker Amendment. So the override of that was a big political issue in 2006, and this is I don't know how to get this up. Huh? What do I do? It's funny, I can't find my little arrow on my...
It's not showing on your computer. It's not showing on your computer, but it is showing up here.
Okay, so how... Oh yeah, okay.
Next summer, I'm going on a camping trip with my friends. On my way home, I'll be in a car accident and I'll be paralyzed for the rest of my life.
In 20 years, I'll have Alzheimer's. I won't recognize my husband or my kids.
Next week, my mommy and daddy are going to find out that I have diabetes.
This is my congressman.
Congressman John Sherwood.
He voted against federal funding for stem cell research.
Is he a doctor?
Is he a scientist?
Why did Congressman Sherwood bet my life that he knows best?
Help me.
Help me.
Who knows? Maybe I'm your mother.
Maybe I'm your grandson.
Maybe I'm your little girl.
How do you know I'm not you?
Stem cell research could save lives. Maybe yours or your family's. Someone you love. Only Congressman Sherwood said no.
How come he thinks he gets to decide who lives and who dies? Who is he?
Majority Action is responsible for the content of this advertising.
So I'm not sure what happened to poor old Congressman Sherwood, but it strikes me as a very sort of violent kind of advertisement, and I hope you agree with me. If you think our politics is bad now, it always was, okay? But here's the thing. The argument from suffering has no bottom. It's a very deep and difficult way to approach a moral issue. And I want to lay that out for you just very quickly, and then we'll talk. So this is an 8-cell human embryo. That's how small what we're talking about is. But nonetheless, it is the beginning of a human life. It's the early stages of human life. I'll tell you a little anecdote. The first day I had lunch, J.K. came to see me at Stanford numerous times. And the first time we had lunch together, he said to me, is the stuff in America about not using embryos, is that just some little fringe fanatic group? And I said, well, no, not really. Our country's pretty evenly divided on this issue. And he said, how can something that small be as important as my little 2-year-old daughter? And I said to him, well, don't forget, J.K., that the argument would be that that daughter of yours started out that small. And that, of course, is the argument here, that the life begins, first stage, and develops, and that you are yourself, in a sense, you're a living human organism at all stages. So that's the core of the conflict over this issue. But I think we also need to be very careful how we think about it, no matter how we approach this issue, whether we agree with that moral principle or we disagree. Because the moral principle itself opens a door that we have to, at the very least, reflect carefully on. And I want to pose 4 questions and just go through them very quickly to show you why I think this is so important and where it might go. So the argument was for embryonic stem cell research, but what more could you do with embryos? Well, as I've already said, a great many things. So that's goes without saying. Will we create human embryos specifically for research purposes? Well, that would be a very useful thing scientifically to do, but does it cross a line for people? There are quite a few places in the world where that's not allowed. That's Rudy Yanish, who created the first transgenic animal. Just to give you an idea how powerful CRISPR is, he told me that it took him 2 years $200,000 and he changed one gene, now he can do basically the same thing in a few weeks for $2,000 and change 6 or 8 genes at a time. So if we do create embryos, how many embryos is it okay to create? One Japanese scientist created 581 cloned copies. And by the way, cloning might come back in as a useful standardized tool to get batches of embryos that are more or less the same. And then we could do really interesting studies with them by some arguments. Where would we get the eggs to do that? Well, that's a little complicated. Some suggest you take them from aborted fetuses. There's something very repugnant in most people's mind about doing that. But Davos Salter pointed out just 10 years ago that eventually induced pluripotent stem cells could be used to make eggs. Then we can make all the embryos we wanted. Said maybe 20, 30 years from now, I'll read in the newspaper that someone made 20,000 embryos and studied their development, and we'll decide it's okay. Of course, the really deep, difficult question that all of us, no matter how we feel about embryo research, must contend with is the question of how far. And it's very, very amazing, because when the principle of 14 days was set in the UK by Mary Warnock's committee, she just died this week at 94, when they set that principle, it wasn't possible to keep embryos alive that long. But more recently, Magdalena Zernicka-Goetz and others at Cambridge and a variety of places have figured out how to keep embryos alive longer. And so the issue of the 14-day rule is being revisited. There was a whole issue in, I think it was Nature Biotechnology a few months ago, arguing that it, from most of the commentators said that it should be extended to 28 days. That's Kathy Nikon, who works at the Francis Crick Institute in UK, and she's doing studies like I've described with human embryos. So how far? Well, my colleague at Stanford, who was on the state ethics committee, said we didn't mean 14 days would be the limit. That could be changed in the future based on what would likely come from neuroscience. Well, what's he talking about? Neuroscience? What does neuroscience have to do with this? Well, his idea was that eventually we'd decide that you're not really a human being until you have some expression of consciousness or self-awareness. And nobody knows quite when that is, but it may not be till well along in development. So other people say, well, since that's the case, then we should use the resources of developing life to not just study life, but to create tissues, cells, tissues, and organs. This is a physician, Julian Salvalescu, who at the time of the stem cell debate said it was morally required that we produce embryos of fetuses for the sake of providing cells, tissues, and even organs for therapy. I debated him at a forum in New York City, and I said, well, how far would you carry this? 60 days, 3 months? And he said, yes. And I said, 6 months? And he said, yes. And I stopped asking. 6 months, we have kids in the intensive care nursery, in the NICU, that are less than 6 months old. So I personally find that really horrifying, but you can decide for yourselves. In the meantime, there's a company in, and by the way, there is evidence that you might be able to use fetal parts for transplantation, not just cellular, but even whole organs. There's a company in Silicon Valley, I guess you had to know that's where it would be, it's actually taking aborted fetal parts and trying to see if they can turn them into transplantable organs. So I just wanted to lay that out for you as the scope of the challenges coming with CRISPR. CRISPR is a fantastic tool, but we have to be very, very careful. Erwin Chargaff was a very major figure in early genetics, but he warned us. Science could very much end up in a morbid ending, the way we're going. So I think I'll stop there, and leaves us a little time for discussing ideas.
Maybe as a first question, you highlighted, I mean, the topic of the seminar is about gene editing, but you highlighted kind of other previous technology developments in biomedical research where there similarly were issues of using technology to cross what at the time were thought to be ethical boundaries, right? And I guess I'm trying to figure out, how do we learn from those? So whether it was originally recombinant DNA technology, or then later you highlighted embryonic stem cells, maybe things like synthetic biology have also gone through this type of cycle. How is CRISPR the same or different from these earlier examples where we had to grapple with ethical issues of pushing boundaries, and then presumably, at least so far, we've gotten by, but maybe that's a subjective opinion.
Yeah, that's a really interesting question. How are we going to figure this out? And it seems like they're coming in stages, and the challenges of each stage are different. I think it's a kind of a progressive scale of freedom and peril, and in that sense, the issues are getting more and more profound and broader in scope. The interventions that we've been able to do thus far into human life have been moderate, actually. I mean, compared to what you can imagine. And germline genetic engineering is I mean, it's certainly a hinge of history that it's happened. I think probably Charlie feels the same way I do about when we were there in Hong Kong. The story was broken the night before the. Is this working? The story was broken the night before the gene editing summit. In fact, the guy who broke the story called me while I was on my way through the airport at San Francisco, and he called me to ask me what I knew about it. And I didn't want to tell him too much because I did know a lot about it. I wasn't sure there had been implantations or babies, but I'd met with JK just a month before, and he told me he had a major paper coming out and that the Associated Press had been there, and so I worried a lot. But when the paper broke, leaked his results, he didn't want to tell before the gene editing summit. But it was a big storm, and everybody was talking about that. It was the topic on everybody's mind. But he was scheduled to speak in the second day, so it was really 36 hours after it was broken. By that time, the press had flown in from all over the world, and about a third of the auditorium was cordoned off for the press. And there were these 4-foot lenses and cameras, and it was very intense. And as he came in, first there was a little mild applause, and then there was a silence, like I've never heard silence. Everybody was focused in on that. And it was just, I think everybody in the room knew we were at the epicenter of the human story for that particular week anyway, maybe for this year or this decade. It's an amazing moment to think that for the first time in 4 billion years of, close to 4 billion years of life on Earth, there's a species now that can turn back and potentially alter its own evolution. And it won't be easy. I'm not one of those transhumanist guys, but I do think it raises very serious issues. I'm not really answering your question, I'm just dramatizing it. I guess all I can say is I hope stepwise we contend with these issues well at each stage so that we're ready for the next ones, because it's going to be a cascade of more and more difficult issues, ultimately converging in neuroscience. And we need to build the foundations of this. This is I think the deepest challenge of our age, and maybe the deepest challenge our species has ever faced.
I thought, hi, I'm Jeff Baker. I'm not at all a real scientist. I'm a historian. And your question, I think, got me thinking some about comparing this moment to, say, 1972. And I think Mike first, to the recombinant DNA controversy of that year that you already referred to. 72 or 73, I can't, when Berg had his moratorium. My question for you, has Rolling Stone done a front page article on this yet?
What did you say?
Has Rolling Stone magazine done a front page article on this story, on CRISPR?
Yeah,
recently. Did it get the front page again? I don't read Rolling Stone. Well, it had a major story on it in '72 that to me just epitomizes how public an issue this was. It was one of the great issues that launched bioethics, as you know.
Yeah.
And it was all over the press. And this has been in the press some, but I guess I'm wondering if it is exciting the level of kind of public discourse and shock. The discourse in the 70s brought up a lot of things that you've talked about, and lots of talk about Frankenstein and the limits of science and what it's doing. And to me, it feels that the culture is reacting some, but not as strongly as it did in that time. And I'd like to invite comment on whether, first of all, you might want to disagree with me about that, because maybe I'm spending too much time doing my electronic health records as a doctor, and I'm not watching the news enough. So you think, is there a difference? Is there a sense that some of our culture is kind of a little less ready to react strongly to this? Maybe part of what's changed is IVF has come in. We are used to embryos, surplus embryos, which was not the case in the early 72s. The 70s were following the 60s. There were a lot of people talking very explicitly about sort of eugenic plans, eugenic purposes of I mean, there was a sperm bank, basically, that had been developed to select Nobel Prize laureate kids and clone them. I'm sorry, not clone them, but to offer them for sperm donors. There's lots of that kind of talk that the 70s were reacting to. I'm just wondering if, in our point, do you feel that that the culture is in a different place? Are we sort of in a point where we just...
That's a big worry. I agree with you. There's a saying from Dostoevsky, man gets used to everything, the beast. And I think it's possible that we get inured to these things, that it's all the more complicated because there's such a mix of positive and negative. And I suppose it's true here at Duke, being such a leading university and research center, but where I live, the whole idea of making the future, of the next new thing, disruption, is very prominent. Everybody's looking for a new way to do something different and exciting, and most of my students really have a dream of starting companies, and even if they're going into the sciences. So there's a very future-oriented view, not a very rooted view of these matters. And I think because there's much positive to be had, and because there's kind of a complexity that none of us are really well equipped to deal with, and because we're all suffering from a little overload from too many controversies, we got kind of moral controversy fatigue. I think you're right. I think that the alarm is not as great as it deserves to be. We're all kind of distracted on things that are, to my mind, less important. I mean, they're important, but these are monumental. This is like, this generation is going to set the direction, set the arrow to flight for the future. And if this generation doesn't think carefully, I mean, right now, you probably know, just 2 weeks ago, there was a call, maybe it was 10 days ago, a call for a moratorium, 5-year moratorium on germline engineering. And there's a controversy about whether or not to have a moratorium. And so, well, what are the arguments for or against having a moratorium? But I don't see much in the way of discussion in the press about it. It might be one of the more important decisions our society faces ever. What do you think, Charlie? Do you sense there's...
Yeah, I mean, it's difficult because I think my world is a very tightly packed, CRISPR-filled bubble. So I'm probably highly biased. I mean, I know, I'm trying to think, I have a slide that's got the cover of Newsweek, The Economist, Wired, New York Times, Wall Street Journal, and all of them have covered, these things at one time or another, with a lot of designer baby schematics on the front page. I can't remember whether I've seen Rolling Stone or not, but so it's definitely out there. I definitely was getting like articles sent to me from my grandparents that they clipped out of newspapers, but I do think, there's probably some amount of fatigue in terms of whether, and it may be also so technical that it's hard for people to wrap their heads around how big of a deal it is or it isn't, which might also be part of why there isn't more scare about it, I guess.
Yeah, I think the technical quality of it, I think, is a big barrier. But see, the problem is that it's conceptually difficult, but actually it's not that hard to do. I mean, JK he was trained in biophysics, and he just found a few people to cooperate with the IVF part of it. And by the way, the story you're reading in the press is not the true story. I mean, it was, what basically happened was it was a big embarrassment to the gene editing summit that he'd done this, because the gene editing summit was partly about science discussing carefully and quietly and regulating itself. And when this happened, the convener of the summit, David Baltimore, stood up and said, just directly, this is embarrassing. This shows we have not been able to self-regulate. But the problem is that if science can't self-regulate, science understands the issues. If science can't self-regulate, how are we going to regulate? And it's a very, very great problem. Because CRISPR is relatively easy to do, it looks like anyway, I think you're going to see commercial fertility clinics all over the place opening up, offering CRISPR, if we don't have some kind of a constraint on it, but I don't know how to constrain it exactly. The story of JK is much more complicated than is in the press. It's not like people didn't know. A great many people knew. And I knew where he was heading. I didn't know that he'd done it. But I was working every time he came to see me, I tried to discourage him from advancing too fast, to slow down. I really wasn't in favor of his project. I told him that. And yet he sort of marched ahead. But he's just a product of what's called Shenzhen speed. Shenzhen is China's version of Silicon Valley. It's sort of Silicon Valley on steroids. There are very many ambitious people pushing very, very hard and very fast to advance things. China wants to be the leaders in the future in biotechnology, and they're not holding back. And a guy like JK got a lot of encouragement both within China and within the United States. And I know the whole story because after the gene editing summit, he called me from Shenzhen numerous times, and we talked at great length. And I know who knew, and I know all sorts of stuff about it that nobody knows. But for the moment, I don't feel I should talk about it. I wish what would actually happen is everybody would talk about it openly and say, look, we didn't kind of appreciate the significance of it or what was happening. But that would be the most constructive thing could happen, is everybody step forward and acknowledge their role. And otherwise, he's going to be the scapegoat and take the whole brunt of it himself, which is not fair. He's a very nice guy, actually. A little too ambitious for sure, and too pushing forward too fast. But I can assure you he was very well-meaning. And as I got to know what he told me, I realized that I still thought he did the wrong thing, that it was really not a good thing at all that he did. But nonetheless, it was more understandable as he would tell me about it. So there's a bigger story back here, and behind it all is the issue of global governance of biotechnology. It's just, these are species issues. We've got to deal with them. If we don't deal with them collectively as a global culture, they're going to be misused somewhere and very dangerously affect the whole of the human species.
I should remember this, but do not. He conducted the editing. He did not create embryos for experimentation, but rather conducted the editing on embryos that were destined for implantation already. Is that
well,
right?
he first did studies of that sort, but then when he recruited patients, and he in this case, the father in the family had HIV and the mother didn't, and there's some social reasons why he believed this was an okay situation to do. They went through informed consent, albeit not probably the kind of informed consent we would find acceptable, but they were not poor, exploited people. At least, I mean, I'm telling you only what JK told me, so I don't know except what I heard from him. But these families were not poor. They were reasonably well educated. They could understand what they were doing. They just really wanted the gene editing. And then he did regular old IVF, but he engaged the CRISPR mechanism at the same time as he made the embryos.
So the intention was to make these two to be born babies resistant to HIV.
Right.
And so you said you think he did the wrong thing. What did he do that was wrong?
Well, Charlie,
Besides get
why don't
ahead
you
of
lay out what he
besides
did
get
wrong?
ahead of the scientific community.
What do you think he did wrong? You're probably more articulate on this than I would be.
I guess, I mean, it's probably where there's ethical mistakes maybe, and then technical mistakes. I mean, I'm only qualified to talk about the technical mistakes, but, and there are many, and I don't want to take up the time to talk about it, but knocking out that one, it's not a gene that ups those babies. Those babies were not, there's other ways to prevent them from getting infected with HIV than to actually do germline editing. So it wasn't a necessary procedure and arguably didn't benefit them in any way. Knocking out that gene doesn't make you 100% resistant to HIV. There's other ways for HIV to get into cells. So arguably even what he intended to do wouldn't accomplish the actual goal of preventing from HIV infection. If they were exposed to HIV, which they didn't need to be, that was a risk that was taken. And then one of the bigger ones is that the actual intended edits in these babies were not made, meaning that and one is that the babies ended up being mosaics. So if you edit the gene when it's a single cell, if you edit both copies of that gene, then all of those edits will get passed on to all of the other cells. But if you inject CRISPR into a single cell and it doesn't edit, and it divides into 2 cells, and then say 4 or 8 cells, and then some editing happens in some subset of those 8 cells, then you end up with what's called a mosaic, where some of the cells in the body have been edited and some of them have not. And even in the same cell, one allele may have been edited and another may not have been. You may even get different editing events in different cells in that hypothetical 8-cell embryo. And it appears, although it hasn't been through peer review or anything like that, but from the data that has been circulated publicly, included what he showed at the meeting, it appears that these babies do not have uniform editing. They have some kind of mixture of somatic cell editing that would either be predicted to not necessarily confer resistance to HIV and or create new versions of this gene that may produce some other protein of unknown function. So there's some technical issues with kind of how all the editing was done and why it was done.
You know, the interesting thing is that I think the major problem has been, for the scientific community, has been the concern about off-target effects. I'd say that's fair. So several people in the conference, I can't remember if you were one of them, but Matt Porteous at least basically said, at least for his work with sickle cell anemia, he felt like the off-target effects were not an issue, that he could do the editing well enough. So JK was going down checking the boxes, and he felt his technology, his expertise was in gene sequencing, and so he felt like he had mastered the off-target effect issue, and he thought he'd checked off the boxes of all the things that the National Academy had suggested, and he read the Nuffield Council report, which seemed to open the door for more liberal use of this technology, and he really believed he was following the general rules. But when he came to Stanford, I kept saying to him, JK, you do this in conversation with the community. This is not a kind of experiment for a private scientific lab. This has got to be a more global effort. And that's what, of course, the National Academies called for. In fact, I went so far as to recite for him the last phrase of the Gettysburg Address, of the people, by the people, for the people. And I told him that the genius of democracy was the collective intelligence of the community, the wisdom that exceeds the wisdom of any one person. And after he left me that day, I sent it to him in an email. I don't know what the Chinese government thought about my sending the Gettysburg Address along, but and when I talked to John Cohen, a writer from Science Magazine, I said, you know, John, more than any single thing, I think this was a failure of democratic process, because had JK stayed in conversation with the larger community, he would have gotten the kind of feedback that Charlie gave, and he would have been cautioned about certain things. And I asked him several times after the summit, did you rush this because you thought somebody else, or did you know somebody else was going to do this? You were going to get scooped? And he's such a nice guy. He didn't say, yeah, I really worried about that or something. That could have been an excuse. He said, no, I didn't think that. And he just was eager for the sake of the patients. He said, the patients need this. And if we talk too long about it, it's going to be another 5 years. And in the meantime, all sorts of people who need this technology are not going to get it. It was sort of the same idea that's going on for mitochondrial transfer. Those of you in science know about that. It's the so-called three-parent embryo. It's more or less pushed forward without general discussion. And same with IVF. It was sort of sprung on all of us. Robert Edwards did 450 implantations before it was publicly known broadly that he was doing IVF. And none of them took until the last one. So what I think we just need to realize above all else, that we're entering a very, very serious moment in the history of humanity, and that the challenges and the opportunities are worthy of very, very serious dialogue, collective dialogue, not just from elite scientists, but from all of those who do the perennial tasks of having the children, raising the children, doing the workaday jobs of life where there's basic competition and so forth. All of us need to be in this conversation. It's a collective conversation for our species.
I'm wondering if you read the consent forms for the participants, and if so, what you made of them, and did you discuss them with JK?
I did not want to get involved in an advisory capacity for him. And I just kept saying over and over, you've got to follow the rules of your country, the regulations of your country, and you should stay in wider conversation with the scientific and ethical communities worldwide. I did talk to him about what constitutes reasonable informed consent, but I have subsequently read his informed consent document, but it was translated, and I don't speak Chinese, so I'm not sure how it came across. But I'm actually, I think he didn't handle that right, but I don't think it's necessarily true that the patients didn't know what they were getting into. I think they did. That's from my understanding. Now, none of us know what we're getting into with germline genetic engineering, if the truth be told, because
It was described as an AIDS vaccination study. You think that's an accurate label?
Well, that's the sort of American cultural translation of what he was doing. And yes, that is sort of what he saw, but here's the background of that, and I'm not trying to justify it, I'm just explaining it. So JK grew up in a small, poor, rural community, a village. His parents were very poor. In fact, he told me that family was so poor that he was embarrassed to ask a girl out that he later married, but he was embarrassed at first to ask her out because her family owned the restaurant. And he grew up in this kind of an environment. He knew the struggles that people go through. He visited the villages in China where there are huge numbers of people with HIV. China had a phase where they believed that somehow or another if you got blood from younger people, older people would gain longevity from it and vitality. And so they were collecting blood from villagers, and I guess the elites were getting the blood, and they went from person to person without sterilizing the equipment. And so they spread AIDS, and there's some villages in China where 30% of the population has AIDS. And by the way, that idea of parabiosis, of using older people's blood, is resurging, and there are companies in Silicon Valley providing blood of younger people again. But they do it hopefully sterile. But these villages with very high concentrations of HIV, JK visited them. He tried to describe to me what it's like for somebody with HIV in China. They often cannot get a job, cannot get medical care, are shunned, and their lives are very, very difficult. And so when he had this idea was if he could break into that by vaccinating or precluding the vulnerability to the virus in these villages, that he could break the cycle of the disorder and help a lot of people. That was his thinking. I mean, I'm not probably telling it as well as he could, but it's not quite the way we would think about it.
You know the status of the babies?
The babies, the last I heard of the babies, and JK did not continue to have contact with them after a certain phase, but to as long as I know, they're healthy. And it's probably, CCR5 is a gene that has subsequently been suggested. There are some studies now that suggest that it affects certain dimensions of neurologic function, in fact, beneficially in some parameters. So it's unlikely that the gene does just this one little thing for this one little portal into the cell, but no doubt it'll affect other things. But there are people with these particular mutations walking around in the general public, so one doesn't know for sure, but it's very likely that he didn't damage these children in any serious way.
But we don't know because these mutations have never been observed before.
Well, they have been observed. 2 or 3% of the European population has the mutation.
CCR5 delta 32, but that's not what these babies got, as Charlie said.
Well, Charlie maybe can answer that better than I can, but I talked with JK about it, and he said that he actually consulted with significantly informed people on this who said that disruption of the producing a stop codon would affect the transformation of the gene enough to prevent the portal from being present. I mean, maybe you know more about it than I do. I'm certainly open to the idea that he did a wrong editing, but
I believe some of the results indicate that he didn't just introduce a stop codon in some of the alleles, but actually removed an even number of amino acids so that you would get instead of just a stop, you'd get like a protein being made that is just missing some chunk of its, I think it was 9 or 11 amino acids or something in the middle. So then you have to start to ask, okay, well, if we just, as opposed to the delta 32 mutation in the Northern European population where it's a premature stop codon, you don't expect the protein to get made. If you just remove 9 amino acids from the middle of the protein, and now this new protein missing 9 amino acids gets made, can we assume that that's not going to have any weird, unpredictable function? And I think we don't know.
Yeah.
The babies are supposedly fine now. So I think another interesting really aspect of this that I'd never thought about before until I read it somewhere after this happened was that everything that we know about the delta 32 mutation comes from the Northern European population. And so introducing that on another ethnic group, assuming that the genetics will behave the same, is something that I hadn't really thought about, but is maybe another possible concern.
Yeah, I raised that issue to him, that you couldn't assume just because the European group responded that way. There weren't compensatory alleles and stuff I mean, there's so many issues, and I'm certainly not trying to defend his science. I'm just telling you what he told me, that he had his reasons. And the reason I'm defending him at all, in a certain sense, is not because I want to defend what he did, but to point out that he had his reasons. And the problem is that so will other people have their reasons for what they want to do. And therefore, we need to figure out a way to govern this so that private reasons don't start ruling, because that's what's very likely to happen. And look what happened with stem cell research. There are many, many stem cell clinics around the world where you can get things that range from the frivolous to the fatal. And there's no way to regulate this stuff. It's out there. And this technology seems like it might be easy enough for infertility clinics to offer it. And boy, will there be a market for it. What do you think?
I don't know. I mean, as you say, he had his reasons, but I think as been demonstrated in the literature and from his peers, those reasons were bogus. And as Charlie said, there are other ways to avoid HIV. This is a gene we understand somewhat, but not completely. It's also very likely that when you mess with it, you make people more susceptible to West Nile virus. I guess, to be honest, I'm surprised and confused that you seem to be more concerned about him and the consequences that have befallen him than to these participants and these babies.
Oh, no, you've got the wrong impression. I'm
Well,
not.
I'm glad.
I'm not concerned. I'm not unconcerned about the babies. I think what he did to the babies was completely wrong. And I'm also, I don't know how to say it any more firmly. I think it's inexcusable what he did. But what I'm trying to explain is that if you want to understand this story, you have to understand how many people consulted with him, encouraged him in the United States and in China, and his rationale for why he did what he did. I mean, for example, I asked him about West Nile virus. He says, there's no West Nile virus in China, and this is not an issue. And that's a fairly narrow view of why a response, because people travel. But still, my point is not to defend JK for what he did. Obviously, he's a human being. We want to be concerned about his fate. Robin Lovell Badge came out saying he might even be executed. Well, I think that would be wholly inappropriate. And if I can help stop him getting executed, I would do that. You know, they executed the head of the FDA in China when he didn't behave properly. And in that case, they said he took bribes, so who knows. But in any case, you know, it's the first principle of Chinese science is you don't humiliate your country. And so I don't want JK to get executed. So in that sense, I'm with you.
I don't think any of us do.
But my big point, my larger point is this, that this is not something we're going to solve by criticizing JK. It's not a problem. He's raised a problem that we have to contend with in a very serious way globally. We can't just say, oh, this was a rogue scientist, this is a one-off, this is a guy seeking fame and fortune. We have to contend with this in a way that helps prevent anything like this in the future.
Last question from Professor Royal.
Hello there. Thank you for sharing your perspectives with us. So the World Health Organization has established a committee to examine these issues of gene editing. I wanted to get a sense of your take. What are your hopes and your expectations of this committee? It's a global committee, has people from all over the world looking at the ethical and social implications of human gene editing.
I don't have the answers, and my expectations are it's
yeah,
going to be a
of
lot
what this
harder.
committee will. You keep talking about this as a global problem, and that we need to think about it globally. And so I wanted to get your sense of what that committee would do, should do. What is your hope that that committee will do? I mean, the National Academies has done its thing in terms of the U.S., but we're talking the WHO, global. How do you think they need to be thinking about these issues as we move forward? Thinking about the pros and the cons as you've laid them out.
There are many issues that need to be addressed here, if that's what you're asking, and I hope they do that. There's a lot more than just scientific risk. Obviously, the risk to the patient is a very serious matter and should be very, very thoroughly addressed. But there are social and personal issues here. So, for example, we want to be very careful not to create a society where there are any kind of pressures to have procedures that people have different opinions on. So, and we're never going to eradicate genetic disease because many genetic diseases arise in development. So we don't want to get to the point where we turn procreation into production. for sure, either just to produce disease-free babies or superior babies. We also want to recognize that with germline genetic engineering, there isn't a patient yet. And so therefore it's an operation on a situation where there are alternative approaches. And for example, my colleague at Stanford, Paul Berg, Nobel laureate that I mentioned earlier, is against germline genetic engineering. He signed the moratorium request, and I talked with him at length a few months ago, and he said that he just doesn't see that the very, very, very few cases in which germline genetic engineering could be legitimately argued for counterbalance the great dangers of introducing it as a medical approach, or beyond medicine even. He thinks that pre-implantation genetic diagnosis could meet those needs. And then there are a whole range of issues associated with the arguments for and against doing it at all. I mean, the first thing we have to figure out is whether we want to introduce it. And then we need to figure out how to introduce it if we do. I'm sorry, it's a very long subject, but those gives you some ideas of what's involved.
Let's thank Dr. Hurlbut again for sharing his thoughts with us.